Have you ever wondered why your parents forced you to eat vegetables when you were younger? Well, today we're going to dig into a paper that explains how your body benefits from eating broccoli and other cruciferous vegetables. The TLDR, you should be eating more vegetables, specifically broccoli. To start, let me be clear that this is not an area of my expertise. I do work with worms and I've read many papers that include methodologies like lifespan assays and RNA sequencing. However, this is not something that I do on a daily basis. So take what I have to say and my opinions with a grain of salt. Always question everything you read and hear. And with that, let's get into it. So, the title of this paper is The Broccoli Derivative Sulforaphane Extends Lifespan by Slowing the Transcriptional Aging Clock. I imagine this might actually be a work in progress. I don't know. We'll see. This is a bioarchive paper, so it's definitely not the final form. I'm not sure where it's going to be published. They don't usually say that in the paper. So yeah, this is the paper that I alluded to in my last video that I'd be going over with you guys. So yeah, let's get into it. So for the background slides here, this isn't going to be things directly from the background. Some of it is going to be kind of alluded to or referenced in the background though. But I wanted to start by just kind of addressing the stuff that we generally know about broccoli. So we've all been told that we need to eat our greens, eat broccoli, eat cruciferous vegetables in order to be healthy, but I feel like a lot of us don't really understand why. So I thought that this paper was both timely and just generally super interesting. And because Monica was one of the lead authors on the paper, it popped into my inbox and I wanted to dig into it. So yeah, let's go a little bit further here. So the key molecule that is derived from broccoli and these other vegetables, but more in a larger quantity from broccoli, is called sulforaphane, and this is the chemical structure here. It's a pretty small molecule, and it's actually derived from a different molecule in the plant. And there's an enzyme that reacts when the plant is damaged. I forget what it's called. Maybe I should have made a slide on that too, but it's not super important to what's happening necessarily. It is interesting though, in the effect that it has when it's cooked. So how they suggest looking into that, and I could make a separate video about that. It'd be a short one if y'all want me to. Just so let me know in the comments if that's the case. But to summarize, there's a different enzyme in the plant that gets activated when the plant is damaged, whether that's through heating or cutting or chewing or anything like that. That enzyme gets activated and breaks down or converts this other molecule into sulforaphane. And that's what that's the active component in our bodies when we digest it. So specifically, sulforaphane has been shown to trigger the activation of genes that suppress cellular stress and toxicity. So the word toxicity was something that kind of like caught my attention because going back to the whole supplement industry and snake oil salesman kind of pitches you see in like infomercials and whatnot, I always had this impression that like supplements that are talking up their anti-toxicity, I guess, effects, I just kind of assumed it was all a load of BS and they're just like, feeding you lies to try to make money off of you. For supplements, that's probably the case, actually. I wouldn't doubt that. But when it comes to bacteria, we actually have very strong evidence that is actually a very positive effect of eating broccoli. Sorry. But yeah, so what happens is this molecule, sulforaphane, gets taken up by your cells, and then there's these other proteins in your cells that are kind of like waiting around to be activated. And I'll go into this a little bit in depth because I think it's kind of important and it's really interesting in general. So I'll kind of geek out here for a second. So NRF2 and KEAP1, when they're not being activated by sulforaphane, are bound together. And KEAP1 is actually, well, so this diagram is a little bit wrong, and I'll explain why in a minute. But sulforaphane triggers the release of NRF1, which goes into the nucleus of your cells and acts as what's called a transcription factor, which basically just upregulates a certain set of genes. And in this case, it ends up being primarily, or maybe not, there's a lot of different genes that get upregulated, but The ones that we're most interested in this paper are the anti-toxicity pathway. And so that's kind of highlighted some of the downstream effects in this review journal here. So you can definitely get some more information on it. But again, this specific interaction here was not quite right. And so what's really happening was detailed in an earlier paper back in 2013, where they showed that sulforaphane actually interacts with keep1 in this SH domain, preventing it from binding with the colon ring adapter, which acts as a ubiquitin ligase for NRF2 and triggers its degradation via the proteasome degradation pathway. So by blocking this interaction, the NRF1 that's bound to keep, or the NRF2 that's bound to keep1, isn't released for degradation. It actually just stays there. So sulforaphane blocks the release basically of NRF2 by the keep1 proteins. And then the new NRF2 that are constantly being produced in the normal cell life, rather than being bound by keep1 and degraded through the proteasome degradation pathway, they're actually free to then go to the nucleus of the cell by transporters and then act as the transcription factor to upregulate these genes that are beneficial to us. So it's a different mechanism, but it's one that is actually very common for a lot of different kind of like quick response pathways in our bodies, or specifically in our cells. I don't know off the top of my head, but I know that there's several other proteins that act in kind of the same way, where some sort of signaling peptide disrupts the degradation of one that can then go and act as a transcription factor. I have a friend who's actually doing some synthetic design of portions of these different signaling peptides and transcription factors as a novel way to treat cancer, basically, with what's called CAR T cells, and basically help the CAR T cells fight cancer cells. It's really cool, actually. So yeah, this is a really, really well studied kind of mechanism, and the literature behind it and the research that's coming out of our understanding of it is cutting edge and really cool. But this is actually something that we've known for the better part of a decade in terms of the specific interaction with sulforaphane and these proteins. So yeah, really cool stuff. Sorry to geek out there, but I thought some of you might appreciate that. So let's move on. So knowing that, we have a couple of kind of unanswered questions. First, what impact does this have at the organismal level? That's what's happening at the cells, but what's happening for our bodies, right? How does the increased activity of NRF2 affect our overall health, especially as we age? So this paper goes over four main, I guess, points. The first being sulforaphane supplementation expands lifespan. And then we'll get into the others in a minute. But this is very common assay done in elegans work, and we call it a lifespan assay. So it's actually one of the easier, I guess, results to interpret from this study. And what you can see in this graph is when animals are treated, specifically adults, with an increasing concentration of sulforaphane. I say treated, but their diet is supplemented. At a peak range of 1 to 200 micromolar concentration, it actually has a lifespan extension effect. So you can see that there's a greater number of surviving animals. We usually use the 50% survival mark as the big delineator. But you can also look at like the terminal point to see like how much it really can potentially extend lifespan. And a normal adult worm will live roughly 3 weeks, so like 21 days. I forget the exact number, but yeah, roughly 21 days. But we can see that this is expanded by like nearly 10 days, so roughly 50% for worms that are supplemented with sulforaphane in their diet. So that's really cool, right? And yeah, this does seem to be concentration dependent, but again, this is pure sulforaphane. So like, as with anything, if you eat too much or consume too much, it can be harmful, right? So you always have to figure out the proper dosage. They also looked at what stage of supplementation the greatest effect was observed. And it turns out that early adulthood in the worms. So day 1 and day 4 had the greatest impact. Although it does look like there's a slightly improved health span as well for day 8 supplemented worms, but it wasn't statistically significant. So the key takeaway is that the earlier you start to supplement with sulforaphane, the bigger the impact. So I think that makes a lot of sense, right? Like as you progress in age, your cells and your body starts to break down over time, and if you can delay that breaking down at an earlier stage, then obviously you're going to have compounding effects. So I think it makes a lot of sense. So the second point they were looking at was the transcriptional profile change or gene expression changes across an organism, and they did what's called RNA sequencing for the whole animals. So basically, like your genes are translated to RNA, and then that gets turned into proteins. So by looking at the RNA specifically, they can see how much the gene regulation is changing. You can look at the protein expression as well, but that tells you less about the gene expression because there's a lot of in between steps that you have to account for. So RNA sequencing is incredibly important and a really common way to test the gene regulation. So this can be a little bit tricky if you've never looked at this kind of data before, and it's not something that I do myself. So it took me a little bit to wrap my head around it, to be honest, too. But essentially, what you have is sequencing files that are massive, and so you really can't plot like this gene is correlated with this gene and do that for like the million or it's roughly like 18 or 20,000 genes, I forget, in the worm. So I could be wrong about that, but it's kind of on that order, like uh, over 10,000, but not more than 20. Um, but doing a one-to-one comparison across all the different genes is an insanely high combination of comparisons, right? So this is a way to analyze all of the gene changes on a one-to-one basis, basically, but in a two-dimensional space rather than a billionth dimensional space or whatever that would be. Okay, so what we can see here is you have two sets of animals. You have control animals that were not supplemented with sulforaphane, and you have animals that were supplemented with sulforaphane at day 1 of adulthood. And then sequencing results were done at these different days after the animals were collected and separated, right? So for the control and supplement fed animals at day 2, they kind of cluster in the bottom left corner like you expect. They're together. Day 4 is kind of similar as well. They're also in the bottom left corner. So this just says that in early adulthood, the gene expression profiles are roughly the same, regardless of supplementation or not. Which, I mean, they're young adults. You kind of expect that there's not a lot of damage accumulated yet at that stage. And then you can look at day 8, and you can see there's a shift away from the early adult life. So this is kind of like midlife. There's a dramatic change here from the bottom left corner to the top left corner, maybe a little bit towards the center as well. But it's obviously significantly different than the early life stage. And then we can see for the day 12, the controls are similar to the controls for day 16. So kind of looks like in the bottom right corner, like an older quote unquote phenotype of expression, gene expression. But we can see that the day 12 supplemented adults are much more similar to like midlife adults than they are to the older adults. And then for the day 16, which is quite old for a worm, the gene expression profile is closer to the older age profile that we see in the controls. Not quite as far to the bottom right as the date, even not even quite as the day 12 is. So there's a marked improvement even at that stage. But basically, this just tells us that there's dramatic changes between early, mid, and late life in the gene expression profiles. So I actually think that's really interesting, even if it takes a minute to wrap your head around how the data is being, I guess, shown here. And again, this isn't showing specific interactions, it's showing across the whole spectrum of genes in the animal. So it's hard to say exactly what's changing, right? Because this is kind of a very macro level analysis. Okay. So using the same kind of treatment where you have day 1 treated or day 1 supplemented worms supplemented with the sulforaphane versus worms that are not, you can see in specific genes a dramatic difference across age, right? So some of these genes, I think, I know GST4 was. I don't remember if the other ones were as well, the anti-toxicity pathway or not. But anyways, the point is that you see a dramatic change in certain genes. So these ones are upregulated in the controls or in the supplemented worms versus the non-supplemented. These LEP418 is the opposite. It's downregulated in the supplemented worms. So yeah, on a gene by gene basis, it varies quite a bit. Based on those genes that were either highly upregulated or highly downregulated when treated by sulforaphane, the authors were able to make a model that could predict the age of a worm based on the transcriptional profile. And so by doing that, they then tested how they fit their model based on the control data, and they were able to show that it was highly correlated. And then when they fed the data from the supplemented worms, you could see that there was a significant, I guess, slowing of aging in those worms, as indicated by the lower grouping of samples. So because this was trained on the genes that were either highly upregulated or downregulated in the treated animals, this is definitely a biased model, but it does fit the control samples quite nicely. So it seems to be an appropriate model as well. But also comparing that to another model that was previously published called bit age, I guess. Um, that model, when fed the control and supplemented RNA sequencing data from the worms, showed also that the supplemented worms in green here, this line, also had a delayed aging phenotype, or not phenotype, I guess in a model, I don't know what that would be described as, but that model predicted slower aging in supplemented worms. So that kind of corroborated the validity of the model that these authors came up with. So kind of tricky because you're dealing with massive data sets and a lot of variables. But if the models work, then there's some validity to it. So it's pretty cool. Okay, so next it went on to identifying what the biggest impact, or when rather, the biggest impacts of sulforaphane supplementation occur. And so they kind of jump ahead here and show a volcano plot of the differences in gene expression, specifically at day 12, because that's when they saw the most genes being affected, which they show in the second part of the figure. But in the first part, you can see that most of the genes that are being affected are being upregulated. There's a good amount also that are being downregulated, but much more of them are on the right here. And you can see, this is the second part of that figure, that of the downregulated genes and the upregulated genes, there are much more, again, than in the upregulated set. And the point at which most genes are affected are at day 12 of adulthood, which is kind of at that border of like midlife to late life. So this is just suggesting that you have a lot of effects of supplementation that are still happening later in life. So I think that that's interesting and useful knowledge, because eating healthy is not just important for kids and young adults, right? It's good for everybody. And then finally, they wanted to link sulforaphane supplementation to the genes that are most affected and most involved in the, I guess, lifespan extension that we're seeing, or health span extension, or both, right? And so going back to this plot, when you look at the genes that are most upregulated, it turned out that most of these were actually in the anti-toxicity pathways. And so they noticed that just by eyeballing it, but they also went and did what's called KEGG analysis, and that stands for the Kyoto Encyclopedia of Genes and Genomes. uh, which will allow. What there's a tool that you can. I think it's free on their website. You can go and um, basically put in sequencing data, RNA sequencing data that you get from some sort of like control and treatment, like they've done and shown earlier in this paper, and that'll tell you that the changes that you're seeing in the expression profiles, it'll tell you what genetic pathways they fall under, right? So this is just an example from one of the companies that actually does this kind of sequence or this kind of analysis. But when we look at the results from this specific paper, this is some of those results. So you can see these are three of the genetic pathways and the specific genes that are associated with those pathways. And all of these are being very highly upregulated. So that tells you that these pathways, which are specifically detoxification related pathways, are all being highly affected by the sulforaphane supplementation. So again, going all the way back to where we started, you're seeing this upregulation of this pathway that is involved in basically like getting rid of things that are damaging or potentially damaging to your cells, or that are just foreign material that can inhibit the efficiency of things that need to be happening in your cell, right? So obviously, really important stuff to help with living longer and living healthier. So the big takeaway is sulforaphane is a small molecule that initiates the expression of genes that will act to reduce cellular toxicity, and that this anti-toxicity boost seems to have a dramatic impact on healthy aging. So we should all be eating more broccoli. That's the big takeaway. One of the things that kind of came to my mind when I was reading this paper is the question of whether or not raw vegetables are better than cooked vegetables. The answer is yes. This wasn't in the paper that we're reviewing here, but I did find it in this other paper in the Journal of Medicinal Food. But basically, the amount of sulforaphane that you can get from raw broccoli, I forget what this was a percentage of, but so with raw broccoli, they were able to obtain 22.8%. The broccoli, which had not been heat treated and had been lyophilized or lyophilized, lyophilized. Sorry, I had to look up this term because I had never heard it before. But lyophilization is basically just freeze drying. So I don't know why they don't just say freeze drying, but that's what it is. Um, that's one of those things that bugs me about papers in general, scientific papers. Like, there's a lot of technical jargon that gets thrown around, and people tend to like not think of non-experts who might be reading the papers and whether or not they'll understand the terms. But I get it because sometimes these terms like they have very specific different meanings from one, like freeze drying and lyophilization or whatever. It might be just there might be one specific difference, or maybe it's a specific apparatus that needs to be used in one versus the. I don't know. So it's kind of on us as readers to take a second to look it up if we run into terms like that, right? But anyways, so from 28 or 22.8% down to 4.2% for broccoli that was heated and then lyophilized. So well, it was steamed, right? Steaming for 5 minutes. So that's a pretty big drop, but I think it's still important that you're still getting a good amount of sulforaphane from that, even after steaming. Which, I mean, steamed broccoli is pretty awesome too. So a lot of people don't like eating raw broccoli because of the taste or whatever, but they also say that mild heating at 60 degrees Celsius, which is like 140 degrees Fahrenheit. Um, I don't really know what the temperature would be like if you're cooking it on a wok or just like boiling it. Yeah, so like you should still retain a lot of sulforaphane. Um, but I guess you can also like if you chop up the sulforaphane and let it sit for a while, then or chop up the broccoli rather and let it sit for a while, then the enzyme that works to make sulforaphane as a result of the damage of the broccoli has more time to act. So you can, if you let the broccoli sit before cooking it, maybe you can get more out of it. I'm not sure, but I think the most important thing is just that like you should eat more broccoli, right? And you're gonna get more sulforaphane. So maybe I'll do a follow up to this, or just put something in the comments to to note, whether the amount of cooking time matters, or what if you chop up the broccoli before cooking, like how long you should let it sit, that kind of stuff. I didn't really bother to look into that prior to making this, but yeah, that's it for this paper, guys. So thank you. All right, if you've stuck around this long, please make sure to hit that like button and subscribe. If you have any questions or comments related to this paper and what we discussed, then please let me know in the comment section below. And as always, stay curious, stay humble, and never stop learning.